Pessoal, boa tarde a todos. Bem-vindos a mais uma sessão do Congresso BRAINN. Eu vou mudar para o inglês agora para apresentar a nossa palestrante de hoje, que agora é a nossa primeira palestrante dessa tarde. So I'm going to switch to English now. So it's a pleasure to have Dr. Adrienne Fairhall here joining us at this conference. I just told her it's a pity that she's not here live, but next time we'll hope she will be able to come. So Dr. Fairhall is a professor in the Department of Physiology and Biophysics and an adjunct professor in the Department of Physics and Applied Mathematics at the University of Washington. She obtained her honors degree in theoretical physics from the Australian National University and a PhD in statistical physics from the Weizmann Institute of Science. She joined the University of Washington faculty in 2004, and now she directs the University of Washington's Computational Neuroscience Center and the University of Washington Institute for Neuroengineering. She has directed the MBL course, Methods in Computational Neuroscience, and she co-directs the University of Washington Allen Workshop on the Dynamic Brain. She has held fellowships from Burroughs Wellcome, the McKnight Foundation, Her work focuses on the interplay between cellular and circuit dynamics in neural computation, with a particular interest in adaptive and state-dependent neural coding. So again, it's a pleasure to have you, Dr. Adrienne. So please, I'll pass you the word.
Thank you so much. I hope my internet holds stable. I had a couple of freezes in the middle there. So thank you so much for the invitation to be at the meeting. And I also feel very sad to not be there in person. I've never been to Brazil, and I hope to come one day. So my lab, as you got the impression from the introduction, is a theoretical lab. And we work by collaborating with a variety of different experimental groups. And I'm going to tell you today about one of the projects that we've been working on, and this one is in collaboration with my collaborator Vanessa Ruta at Rockefeller University. So let's move on. So dopamine is probably the most publicly famous molecule in our brain. Many popular articles talk about dopamine and its role, that it's obviously implicated, right, in providing us information about desires and about rewards. So at least that's the popular image of it. What do we know from neuroscience? What does dopamine do? In fact, dopamine has many different roles, as many of you probably know. Certainly, it is implicated in reward. This is an old experiment that shows after the application of amphetamine, you get a big burst of dopamine and nucleus accumbens and in the caudate. So it clearly does signal stimuli that are rewarding. More commonly now, it's thought about rather than reward necessarily as value, giving information about value. But dopamine also conveys information about many other things. It's implicated in movement. It's implicated, as we know from reduction of dopamine in Parkinson's disease being very critical for the onset of that movement disability. It's involved in action selection, and more recently, it's also been implicated in motivation and in informing kind of the vigor of movement. From a theoretical point of view, as a theorist, we are very interested in dopamine because it is being deeply involved in many models of learning, and in particular of reinforcement learning. Reinforcement learning is an algorithm that is believed to be a biologically plausible way for organisms to learn, right? So you have an actor in the world who generates some behavior. And so we can think about it as sort of experimenting. It generates different kinds of actions that interact with the environment. And the system monitors itself, or there's something about the system is acting as sort of a critic of those actions, and then giving information about whether or not a given action caused reward to the actor. And so through a continuous feedback loop between action and reward, an actor can learn to behave in a way that carries out a desired goal. So what's dopamine's role in that? Famously, Wolfram Schultz's lab a few decades ago made the argument that dopamine was acting not necessarily as reward directly as people had thought, but that it conveys signals about reward prediction errors. So here's the experiments that he based that on. Here's an example where an animal is receiving a reward at a particular time, and you see an increase in dopamine firing after that reward. Now, if you train the animal such that when you present a conditioned stimulus that will predict the reward in the future, eventually the dopaminergic responses will happen after the conditioned stimulus. And so the reward information, the reward prediction is really happening not at the time of the reward itself, but at the time of the cue. And so the dopamine neurons fire at that point. Now, if you have an animal that's trained to associate that conditioned stimulus with the reward after some time, and then on some trials, the reward doesn't happen, so it sees the conditioned stimulus, responds as though a reward would come in the future, and then the reward doesn't happen, you see now a dip in the dopamine activity. And so these experiments were thought to support the idea that dopamine acts as a reward prediction error. Now, an interesting place to look for evidence of what dopamine is doing in learning is in Drosophila mushroom bodies. So these are a small localized area within Drosophila's brain that have been implicated for many years in associative learning. And so it's sort of a tiny compact evidence of the role that dopamine is playing in learning. So here, we're going to zoom in on the mushroom bodies, and we can see something about their structure. So they are divided. So they consist here of a calyx, which contains about 2,000 Kenyan cells. Kenyan cells convey information about olfactory signals into the mushroom bodies. And the mushroom bodies are divided into multiple compartments, about 15 compartments. In this talk, we're really going to focus primarily on these four, on the gamma lobe of the mushroom bodies, primarily because those are ones we will discuss. All right, so let's have a look at sort of the architecture of the mushroom body. So we can simplify it in this picture. So these Kenyan cells that I mentioned carry dispersed and distributed odor information throughout the mushroom bodies. So they have this sort of projections that innervate all of the compartments, so that information is distributed throughout the mushroom body. We also have mushroom body output neurons. So the mushroom body then sends information out to guide movement in response to these inputs. Now, the key part that we're going to be talking about today is that they also receive inputs from dopaminergic neurons. Different dopaminergic neurons innervate particular compartments, and they act at the synapse between the Kenyan cells and the mushroom body output neurons. And so they are extremely well placed anatomically to play the role of a modulator, or something that can help to evaluate the relationship, right, between the inputs and the outputs, and to change the strength of the synaptic connections that relate input to output. So Vanessa Ruta's lab has previously shown that they can indeed play that role, that this dopaminergic network can play the role of a kind of dial, a switch between the strength of the signals coming in from Kenyan cells and the mushroom body output neurons, acting in ways that are fast, selective, so individual compartments can be independently controlled, and reversible. And that provides, in principle, a substrate for the animal to exhibit behavioral flexibility as a function of the information coming in through the dopamine network. So that raises the question of what information is in fact contained in that dopaminergic activity. So one beautiful experiment that really initiated my collaboration with Vanessa was this one. And so Vanessa's lab opened the fly's brain and are able to image these 4 compartments of the mushroom body, the gamma 2 through gamma 4 compartments. And here they are doing that while the fly is receiving different kinds of stimuli. So in the first case, they're receiving a sucrose reward, and in the second case, receiving an electric shock. And consistent with so what you're seeing here is a calcium signal from a calcium indicator that has been expressed in the synaptic terminals of the dopaminergic neurons. And so we're able to understand how the dopamine signals are coming into the mushroom body by imaging from those dopaminergic terminals directly. So in the case that they're receiving a reward, they do what, classically one imagines the quality of that reward. So compartments 3 and 4 here are, 4 and 5 here are responding with a positive response to that onset of the sucrose, whereas the other 2 compartments, gamma 2 and gamma 3, are suppressed when that happens. So in the opposite case, when now the stimulus is negative stimulus, here the gamma 4 and gamma 5 compartments in this case are suppressed, while the gamma 2 and gamma 3 compartments respond with a positive signal. And so this seems to support the very straightforward idea that dopamine in this case is carrying information about reward, positive or negative. There is a little bit of a twist in that, though, and that was realized by the lab when they noticed that here, we see these strong signs of the valence of the reward. But when they were imaging from the fly when it was not receiving either a positive or negative reward, when it was, for example, still, they saw a kind of shadow out of that same pattern of activation, weaker, but still very much there, that gamma 4 and gamma 5 were reacting with a positive signal, gamma 2 and gamma 3 negative, when the fly was still or enacting the behavior that it would undergo when it would stay still and consume the reward. And in the opposite case, when the fly flailed in the way that it typically did in response to the electric shock, they saw the opposite pattern. And so this really raises the interesting question of what is in fact driving the dopaminergic activity. Is it really the valence of the reward, or might it be related to the movement that the fly is making in response to that reward? Okay, so here's a little movie of the experiments. So now what we would like to do is, rather than just do this very simple protocol where the fly gets given a stimulus and we see what it does, now we'd like to investigate how it is responding in a more naturalistic case when it's actually moving. And so here the lab put the fly on a ball, and the ball has texture on it, so that through using video imaging, you can track the movement of the ball and thus extract the fly's fictive trajectory in this sort of virtual reality environment. And at the same time, it's head fixed, and so one can do the imaging that we saw earlier, and now you're watching in real time how the dopaminergic compartments, let's just run that again if we can, how the dopaminergic compartments are responding while the fly is walking. Oops, let's have a look. Okay, and so they additionally put the fly in conditions with airflow, and as we'll see later, we can add to that puffs of different odorants and see how the fly responds to those puffs of different odorants. Okay, so this is the result of the experiments. We see we can now extract the fictive trajectory and evaluate the fly's movement at the same time as monitoring activity in those 4 dopaminergic compartments. In this case, while, for example, a burst of apple cider vinegar is being injected into the airflow. All right, but let's just start with the simpler case where now we're back in the event where the fly is ingesting sugar. And let's have a look at the dopaminergic signals during the sugar ingestion. And you see the same effect that I showed you earlier, large response from gamma-4 and gamma-5 compartments for the sugar ingestion, suppression of gamma-2 and gamma-3. But what I wanted to show you is what happens while the fly is walking around, enacting movement, right, on the ball, away from the time at which it's ingesting the sugar. And so in black, in each case, is the trace of the forward velocity that's extracted from the frictive trajectory, and overlaid are the fluorescence patterns from the 4 gamma compartments. And so what I want you to notice is, here's the strength of the responses during the sugar, but you can see that what's going on at all the rest of the times while the fly is simply moving with no sugar reward at all, those signals are comparable in size to what's happening during the experience of the rewarding stimulus. And these 2 compartments that were suppressed, in fact, during the experience of the reward, have vigorous large activity at all times that, as you can see already by eye, is quite correlated with the movement. All right, so let's evaluate those relationships during free movement. So the very simplest evaluation that we can do is simply to see that the DANs track movement even in the absence of odorant. Right, so here in red are times when the fly is not moving, in green are the times when it's walking, and this is the signal that we extract from gamma-3 in this case. So you can see that there's pretty much a perfect correspondence between the fly's state of movement, walking or not walking, and the response of the gamma-3 lobe. So we can look at gamma-2, gamma-3, and gamma-4 together, and you can see again that there's tracking of in all of those 3 lobes to some extent of that transition from walking to not walking. So if we try to predict the walking state from the responses of the gamma lobe, if we just use gamma-2, so let's just go back, gamma-2, here you can see it's tracking it okay. Gamma-3 is clearly doing a great job. Gamma-4 maybe less so. And so that shows up, you can see that very clearly in this confidence in our prediction. So gamma-3, we can predict the walking state almost perfectly with probability 1, gamma-4 less well so, gamma-3 somewhat less so, but if we combine all 3 of them, we can get to almost 95% accuracy in predicting whether the fly is walking or not. And so here, a little teaser maybe of an issue that we don't really fully understand yet, but just to show you because it's very interesting. We can have a look at those relationships, right? Those coefficients of how well the motion is correlated with the gamma activity in different animals. So for every single animal, that probability of gamma-3 being correlated with movement is very high. And the rest of the cases are organized in order of the strength of the correlation between walking and gamma-4. And what you can see is that from individual to individual, there's quite a bit of variability. They, generally have a strong relationship, but it seems to be somewhat anti-correlated. So gamma-4 for some flies is very anti-correlated. For other flies, it's very correlated. When it is anti-correlated, gamma-2 is very correlated. And, there seems to be a kind of reciprocal relationship between gamma-2 and gamma-4's ability to encode that variable. And we'll have a look a little bit in more detail at that data later, but at the moment we don't quite understand what that means, that level of individual variability. So here you would have noticed, I think, that although gamma-3 is clearly encoding kind of this on-off behavior of walking and not walking, there's a lot more going on in the temporal fluctuations in the gamma lobe activity that you can already see by eye here, clearly does seem to track variations in the movement. And so we can evaluate that correlation. So one way to do that is to compute the linear filters that relate the gamma activity to the time-varying forward velocity. So how we compute these is we simply cross-correlate those two functions. And what that gives us is a set of filter values acting on, here in this case, forward velocity or on angular velocity, right? We can choose which components of the movement to analyze. And what these filters tell us is that if we want to predict the gamma, the DAN activity at time t equals 0, these numbers in the filters tell us how to weight the forward velocity temporal trace in order to make that prediction. And so what is particularly important to notice here is that the dopaminergic activity depends primarily on events that happened in the forward velocity going back into the past, right? So if you want to predict what's happening in DAN activity right now, what it depends on is this weighted sum, according to these filter values, of whatever was happening in the fly's movement in the past. And that's important because you remember I mentioned to you that dopaminergic activity has been thought to be implicated in, say, motivation, right? So in that case, one might imagine that dopaminergic activity might predict what the fly is going to do in the future. Here we see very clearly from these filters that the dopaminergic activity really depends. It's reporting, right, what happened in the past, what the fly has already done. That's the information that's coming into the mushroom body lobe. So there's a little bit of decay, you can see here, after 0, right? So that's weighting what's happening for some time in the future. But that degree of decay is just consistent with the amount of intrinsic correlation in those signals. You can't get rid of that because the signal of dependence going into the future. All right, so now let's have a look at the values that we get for correlations between the gamma lobe activity and movement. And for gamma 2, we see fairly good, strong encoding of forward velocity. Same for gamma 3 and same for gamma 4. Gamma 4 is probably the strongest correlation between gamma lobe activity and forward velocity. And they also somewhat, more weakly encode the angular velocity. So here these traces are predicting, right, the gamma lobe activity from the forward velocity by applying those filters. So we take the forward velocity, we run them through those filters. That gives us a prediction for the gamma lobe activity, and you can see that we get reasonably good agreement. Not fabulous, right? If we quantify that in terms of the proportion of the variance of the signal that we can explain, you see that those numbers are modest, right? We're not getting 100% of the variance. These signals are very variable, but we're capturing, maybe 30% of the variance. Okay, so everything we've just been talking about was when the animals were moving freely, but as I mentioned to you before, the fly is in a condition in which it's receiving airflow, or it can receive airflow, through some really beautiful engineering from Rafi in Vanessa's lab. What now the fly can experience is that they have built a closed loop version of this arena, in which, as the fly moves, one is able to track the fly's movement using the ball and counter-rotate the pitot tube that's delivering the airflow. And so what that allows them to do is to have the animal sort of moving in a true virtual reality airflow environment, so that the world is changing in response to the animal's own movement. And so now in those conditions, in closed loop, the animal walks in this airflow in a fairly random way. But you can see that when it gets a puff of apple cider vinegar, which is an attractive odorant for Drosophila, it tends to orient into the airflow, right? So you can imagine that if you get a puff of an odorant that's attractive, you would walk upwind in order to try to reach the source of that odor. And that's what you see this animal doing. So it doesn't quite turn directly upwind, it turns at some slight angle toward the airflow, but tracks upwind into the source of the odor. And so we shouldn't be surprised to see that the dopaminergic neurons do track the onsets of these appetitive odors. And so now these odors are being applied at random-ish times. And so, these the gray bars here are when the odor is being applied. And this case, gamma-4, gives a nice strong response to that appealing odor. And again, if we do the same analysis that we talked about before, we compute a filter that relates the gamma-4 activity to the odor. We do see a nice clean filter, and overlaid here are the filters that are calculated for many different animals, and you can see that the shape of that filter is very consistent from animal to animal. So some of you might be thinking that we've just seen that gamma-4 signals information about movement. When the odor comes on, the fly tends to move in a certain way to respond to that odor. And so what is odor and what is movement? So, we can see here a very nice example where in this example, the odor came on and the fly tracked upwind toward the odor. And in the, so here's the heading, right? The heading stays fairly constant during that time. And the forward velocity stays small, right? So that the angle relative to the airflow is small, and also somewhat increases its forward velocity. And nice big signal from gamma-4, right? Which might be due to the odor or might be due to this movement. In this case, the fly didn't move toward the odor, right? Instead, it turned orthogonal to the odor for some reason. You can see that from the direction, right? From the heading, it's not down here at 0 degrees. It's, at 90 degrees, close to. And in this case, no response, right? From the gamma lobe. So that suggests that, possibly what we're really seeing here in the gamma activity is primarily an encoding of the movement of, in this case, the change of heading. And if we just look at all the events that happen during the application of odor, we see a strong correlation between the response of the gamma lobe and the change in heading. So ultimately, one would like to build a model where one incorporates multiple influences on DEN activity. I think it's, in our hands, we're not able to eliminate, the possibility that there is directly response to odor. There certainly is, but we need to also incorporate in our model a dependence on these additional variables, the heading and the velocity. And so what we do is to build a kind of combined model in which the odor is filtered, right, through some linear filter that we're able to extract from the data, but in combination with information that's being filtered from about the animal's heading, and also it's about its velocity. And then we combine those three factors together in order to generate a prediction from for the DEN activity. So one interesting issue that we came across is that the tracking behavior is context dependent, right? So what I mean by that is when the fly is moving in low airflow, it tends to do these very convoluted, trajectories. It sort of does little loops and turns around. And then, as we've seen a few times, when the odor comes on, it tends to turn upwind. That, right. So here we can see the responses of the gamma lobe, of the gamma activity in these cases. That is different than what happens in high airflow. So in high airflow, it seems to be aversive for the animal to walk crosswind, and so it tends to just track directly upwind. And so here's an example of a fictive trajectory. It just continues to move right at a slight angle with respect to the airflow. And now what happens when the appealing odor, the apple cider vinegar is turned on, is that the fly, rather than reorienting, it tends to just walk faster. So it clearly responds to the odor, but it does so by moving more rapidly toward the source of the odor. So here's the heading, right? It stays relatively constant. because it's already going in that direction, but the forward velocity takes an increase. And even though, before we saw the heading is strongly encoded by gamma 4, now we still get a large gamma 4 response, and that seems in this case to be correlated with the forward velocity. So we can look at the encoding of those different signals of the average heading and the forward velocity. And in low airflow, there's strong encoding of the average heading, somewhat weaker encoding of the forward velocity. In high airflow, there's no significant relationship between the average heading, even though there is quite a lot of variability. So if there was a relationship, we should be able to see it. Whereas now there's a much more significant response to the forward velocity. So context is influencing the information that's being extracted. If we go back and look at this more fine-grained representation of what is being encoded in those signals, in terms of the filters, we can extract the filters under low airflow conditions and under high airflow conditions. And you also see a change there, right? So in the low airflow condition, there's almost no, quite, relatively, well, okay, so it goes the other way. So there's a clear filter in the high airflow of the heading, although we're not seeing a very strong relationship between it. But the shape of these filters is being influenced by the change from low to high airflow. Here we see a very different encoding of both of the in gamma 4 of the heading and of the forward velocity instantaneously. So here's I won't actually spend much time on this. It's just to pull out all the different variables for all the different compartments, and there you can see across the board there are changes in the slope and in the nature of the encoding for all of those variables. So how can we understand this? What we realized was that what may be an explanation for these results is that there is potentially a conserved variable here. So although average heading is encoded differently, forward velocity is encoded differently, what seems to be encoded the same way is the degree of upwind displacement. That is, how far the fly moves in the direction of the odor. So, in one case, it's using a strategy of reorientation into the airflow to make that movement upwind. And in the second case, it's instead, since it's already oriented upwind, it's instead moving faster. But that change, right, that parameter of upwind displacement in both cases is equally strongly encoded. And so what seems to be possible is that what we're really seeing in the dopamine signal is rather, in both cases, we're able to decompose the movement into these different movement parameters. Maybe the key issue is the fly's movement. It's having done some movement that moves it toward a goal that is triggered by the presence of the odor. So in low airflow, the change in heading predominates. In high airflow, the forward velocity predominates. But in both cases, dopamine serves to signal movement potentially toward the goal. So we also saw some additional information embedded in these dopaminergic signals. And everything I've told you about so far is in a paper that was recently published in Nature Neuroscience. This little piece didn't make it into the paper, but I wanted to tell you about it anyway, because I think it shows even more of the richness of the representations. So here, what you're seeing is that during these experiments, both sides of the mushroom bodies were imaged. And so all 8 compartments from the left and the right. And we also looked to see if there was any interesting information that was being conveyed sort of differentially across the two lobes. And what we found was that it appeared, the difference in gamma 4 activity in particular appeared to correlate with turning. So there's something of a correlation here with respect to gamma 4 and the angular velocity. But then we realized that this, in fact, is a stronger correlation, a better correlation with the wind direction. So turning and wind direction in this prep are quite strongly related to one another. So the better way to think about it really is in terms of the air, the angle of the airflow. And so if one computes a filter between the difference in the gamma-4 lobes and the wind direction, you see this very clean. Well, this one, that there's some fitting issues that make this oscillation. You should just smooth that in your head. So, but clean, clear filter that is also reproducible from animal to animal. So what you've seen is that dopaminergic neurons are sort of instantaneously multiplexing multiple variables. They all, as we saw in the beginning, contain information about reward context, but they also tell the fly about walking state, about the presence of an odor, and they track movement variables during odor, and also convey information about this sensory variable, about the wind direction. So one of the roles of the mushroom body, or at least some previous papers, and this paper by Ilona Caro, I forgot the second part of her name, her lab has shown in the past in collaboration with Juliana Georgieva, they did a similar experiment, similar set of experiments with much sort of lower resolution. So they were imaging across the entire mushroom body with all the 15 lobes, all the 15 compartments. And they saw information about many things, including about some things you might interpret as internal state. So in this case, the fly was put in conditions in which it was starved and hungry. And they saw that some compartments seemed to signal the hunger, right? The internal state of hunger, including gamma-4, right? So there's some response here that seems to signal something about that internal state of hunger. So we were interested to follow that up in our experiments and to see whether, as we saw for behavior, right, that something about the environment changes the animal's context. Is that true also for internal state, right? And so one thing one always has to bear in mind, and what we saw from our experiments, is that context, it could act directly to change the coding properties, but it could also change the behavior. And then the behavior is being encoded through the kinds of filters that we saw already, and that will change the responses of the dopamine neurons. And so it's important to bear in mind that either thing could happen, right? You could directly encode something about the internal state, or what you might be seeing is a reflection of the behavior that changes as a result of the internal state. And that is what we discovered happens with respect to hunger. So when the fly is starved, it behaves vigorously, right? It moves vigorously toward the attractive odor. Whereas when it's fed, it doesn't tend to walk vigorously upwind at all. It sort of does these more convoluted trajectories. And then when the odor is switched on, it barely responds. And so it does not respond particularly to the presence of the odor. And so what we were able to demonstrate was that under these two conditions, the encoding properties were actually the same. It's just that the span of behaviors changed as the fly changed its responses under those different behavioral contexts. So, the starved animal moves vigorously, the fed animal does not move vigorously, but if it were to move as vigorously, even while fed, it drives the gamma-4 activity to the same extent. So, as I mentioned before, dopamine has been thought to be implicated in motivation or vigor. And from the filters that I showed you, we didn't see any particular evidence to suggest that dopamine is telling you anything about what the fly was going to do in the future. However, some causal manipulations that we made do seem to suggest that there is a role as well for dopamine in invigorating behavior. And so in these experiments, we had flies that had optogenetic channels expressed in the dopamine neurons, and now they can walk in a chamber like this. One can shine light on that activates those channels. So here's multiple trials where the animals are simply walking, and an odorant comes on, and during the presence of the odor, they walk upwind. In this case, they were exposed to light that, in this particular case, inactivated the dopamine neurons. And on that trial, you see that there's a significant decrease in their willingness to walk upwind. So, just to show you some examples. So here's, there are quite a few different Drosophila lines in which different subclasses of dopaminergic neurons can be manipulated. So here, one particular class that innervate the gamma 4, 5, 3, 4, and 5 compartments. When those are inactivated optogenetically, the fly's upward displacement decreases in starved animals. Same for this other type of dopaminergic line that specifically innervates the gamma 4 compartment. You also see a decrease in the upward displacement. If instead they're activated using a different channel, you see in odor and in fed animals that don't usually respond, right? When the odor comes on, there's an increase in their willingness to move upwind. So that is it's suggestive that dopamine does in fact play a role in invigoration, and we do have some shadow of evidence in our data that is true, not just under these extreme conditions in which the neurons who are activated or inactivated optogenetically. And so if we go back to our analysis and we look at the correlations between the dopaminergic activity and movement parameters, forward velocity or heading. And now I'm going to sort of zoom out on the conditions that we looked at before. So before I just showed you the filters that relate instantaneously movement to dopaminergic activity. So those filter values show up here along the axis. So these are cross-correlation matrices between movement and dopaminergic activity. So those filters are the values that occur here along the diagonal. So we see, we reproduce those at this much larger scale. But it turns out that if we look out to more extended times in the future, we do in fact see correlations between movement and dopaminergic activity that are in the future. So anything here below the diagonal is in the past, is sort of causal, that dopamine is conveying information about things that happen in the movement in the past. Anything above the diagonal is the future. And so we do, interestingly, see some reflection of correlations in the future. Not quite sure what to make of this because of this, somewhat weird block structure. So one worry is that maybe that's sort of an artifact, that when the animal starts to move forward, right, it continues to move forward in the same way over a long period of time, and that just generates sort of spurious correlations. But we computed the autocorrelation of the movement signal itself, and that has quite a bit shorter timescales than what we're seeing here in these correlation patterns. And so we can't simply rule it out on that basis. So perhaps some supportive evidence that there is a role for dopamine in promoting behavior. All right, how am I doing on time? I should wrap up. Yeah, so I wanted to just point out a little bit about those interesting correlations that we saw before. So what it appears, so we're looking here at a period where the animal is moving, right? And looking at instantaneous correlations between the movement and these different gamma compartments. And these colors sort of tell you the strength of that instantaneous correlation. So what we saw, what seems to be the case, is that there are times when it's very correlated and sometimes when it's somewhat less correlated. But what is always true is that the gamma lobes are correlated with one another. Right? So there's correlation with the outside behavior, but there's also quite strong correlation sort of internally among the gamma lobes. And I kind of pointed that out a little bit from fly to fly, but it's also true within an individual animal. And so these are responses from gamma 2 and gamma 4. If you have great memories, you'll remember that, for different flies, the relationships between gamma 2 and gamma 4 seem to be sort of reciprocal. We see that also at the level of an individual fly. And so here, what you're looking at are responses from many events in which the fly initiated a movement. It starts to move. And during some events, the gamma 2 activity is very high and the gamma 4 activity is very small. But there are other cases in which for the exact same movement or exact same class of movements is onset, gamma 2 doesn't respond at all and gamma 4 responds very strongly. And so, and here, gamma 3, as we saw at the very beginning, very strongly encodes movement onsets. That's very reliable. These two, gamma 2 and gamma 4, are much less reliable about the way it encodes that movement parameter, but are very reliable in the sense that they are always kind of anti-correlated with one another. And so one can kind of pull it all together in a picture like this, which is that the different gamma lobes are getting information from dopamine signals about those many properties that we already talked about reward valence, about the walking state, the walking speed, and even the wind speed, if we take into account the other gamma lobe as well. But they also interact among themselves to generate these internal correlations. And the role of that, we really don't understand much about at the moment, why that might be a useful strategy for the circuit. But to zoom out even further, what I think this forces us to do is to think a little bit more deeply about what computations are occurring in the mushroom body. There's a very nice picture, right, that you take an input odorant, you want to generate a behavior in response to that odorant, and the DANs would tell you if it's a good odor or a bad odor that you would strengthen a certain type of behavioral response to that. Instead, what we're seeing is that these DANs are containing information not just about the valence, right, the goodness or badness of that odor, but about many other things, about self-motion, about the stimulus context, what the airflow was doing at the time, as well as about the reward context. And so it really forces us to think about different kinds of learning models in which all of that information can be used, right? So in this original idea of dopamine as reinforcement learning, maybe all of this information about state and reward are all being delivered, not just the property of the reward by the dopaminergic signals. All right, to summarize, what I've shown you is that dopamine signals convey rich content to the mushroom body, and that sort of challenges simplified models of their role in learning and makes us think perhaps more deeply about how such a network learns and computes and enacts flexible behavior. I've shown you that dopamine encodes instantaneous movement in a context-dependent way, and that's consistent in a way that's consistent with encoding movement that is satisfying a goal. And finally showed you that understanding the role of state dependence, hunger or behavioral context, requires accounting for correlations between state and behavior. If different states cause you to behave differently and your system can be explained by simply encoding behavior, then that is perhaps a simplified way of understanding what state really means. All right, so I'll stop there and thank particularly my team here. So my wonderful collaborator Vanessa Ruta and her lab, Rafi Cohen and Ari Zolan, and my former student Rich Pang, who was really the lead in doing all of the analysis in this project. So thank you. I'm going to unshare so I can see people, and hopefully you won't need me to go back to different slides. But if you need to, I can share again.
Thank you so much, Adrienne, for this interesting talk. It's quite different from what we do at the group here. I am not able to turn on my camera for some. Wait just a moment. Alguém pode me ajudar? Não estou conseguindo abrir minha câmera.
Rafael, pode ajudar a professora, por favor?
Sim, estou solicitando aqui, só um minuto.
Eu não sei o...
I hope the video, the connection stayed relatively stable through the talk. I got a few notices that it was unstable, so I hope that didn't interrupt it too much.
I'm trying to start my video, but I'm not able to. So I don't know if I just...
Doutora Gabriela, nós estamos na tela com a outra professora. Pode fazer as perguntas, a gente deixa lá em tela cheia e a gente vai solicitando por aqui, tá ok?
Tá bom, vou fazer as perguntas. So, Adrienne, I'm gonna apologize, but I'm not able to switch on my camera, so I'll just ask the questions without you seeing me. I'm sorry about it.
I'm sorry too. Nice to see somebody.
Yeah, I know. It's okay. So, you said at the beginning of your talk that dopamine is responsible for enabling movement, and I was wondering, because we work here with rehabilitation of motor rehabilitation of patients that have lost movement due to different things, but mainly stroke. So how is your work in that way could help? You mentioned something about more rehabilitation at the beginning of your work. Do you think your work could help somehow to design rehabilitation strategies for?
Yeah, that's a great question. So I guess the answer to that really comes down, another way, another route into that question is the issue of interspecies comparisons, right? So we work in this project on looking at the role of dopamine in Drosophila. My lab also thinks about what dopamine is doing in birds, right, which has actually a very comparable basal ganglia, cortical, thalamic structure. And why I find these types of experiments interesting and useful is that by looking at these sort of reduced models, right, Drosophila is a very, it's a very compact version of a learning circuit. Birds are, again, maybe a slightly more sophisticated model. They're much more similar to a mammalian model, but in song in particular, they're only learning the one song, right? So it's, again, a very stripped down circuit. So what I hope from our study of Drosophila and of birdsong is that we will see principles, right, for what's going on with the use of different strategies in learning, with the use of dopamine as a molecule that can help us to think perhaps in a more abstract way about how to, what's going on in a mammalian system. And I remember when I first gave this talk where we showed these very complex representations in dopamine in the fly. Ilana Witten, who is an investigator at Princeton, came up to talk to me after the talk and said, "Oh, we're about to publish. We have we see very similar things in the basal ganglia circuitry in mammals, that they also saw very diverse representations of information." And that, somehow what had previously been sort of thought about as just a reward signal indeed did, they were intermixed with movement signals and other kinds of signals. And so where I hope that we can make an impact is, identifying this complex representation and thinking about what it tells us about a learning model. And so what Ilana and her colleagues have started thinking about, and I think our work really points to that as well, is thinking that perhaps reward is this very multi-valued thing, right? So when one performs an action in the world, and there are many ways in which that action is rewarded. One in which you carry out the movement and you achieve the success, right, of having done the movement. as well as when you get some good thing like a food reward or whatever in response to that. And so that's where I think one can make deep connections is looking at simplified circuits, seeing what the principles are, and then trying to apply it in a broader case. The question of motivation and whether one can sort of stimulate to motivate behavior, I know is something that many people in the movement field are starting to think about. The work of Risa Shadmir, for example, at Johns Hopkins. He's very interested in the idea and has been really laying out some very clear evidence that vigor and motivation are a key role for dopamine. And I think he's definitely has a lot more interesting and targeted things to say about that than I would be able to. In our hands, we're really trying to learn about sort of the fundamental models. I think in order to bridge the gap to rehabilitation, you really need to work in the human context, right? To be able to map out those relationships in the human brain. All right, very long answer, perhaps not very precise either, but. Were there any other questions?
Professora Gabriela, você nos ouve?
Yeah, I think we might have lost her. Gabriela, are you still there?
Thank you, Dr. Adrienne.
Okay.
Thank you.
Great. Thank you so much for having me.